Yes, I'm going to talk about multiscale structural characterization by an emerging technique, I would say, called scanning small and wide-angle scattering imaging that you can get access to via the remade project. So what is MAX4? It's a national research infrastructure in Sweden, Swedish national research infrastructure that is located in Lund in southern Sweden. And it's funded by both public and private money, including a large number of Swedish universities. Without going into details, basically it's you could say it's a versatile X-ray microscope where external users or researchers can come to carry out experiments to understand the materials they're working with. Mainly then in material science, physics, chemistry, and biology, biochemistry. And we are open to external researchers from all around the world. Now, if we look at the map of schematic map of MAX4, it looks roughly like this. So we have two circular electron accelerators. We accelerate electrons very close to the speed of light. They emit synchrotron radiation, and then we use that synchrotron radiation to study materials. And with the energies that we operate these accelerators, we reach up to X-ray energies. And then we can do X-ray scattering, diffraction, X-ray imaging, X-ray spectroscopy. And we're currently operating 16 instruments, which all differ in terms of the X-ray techniques that they provide and the kind of science they are suited for. Now, for those who are not familiar with synchrotrons, there's a very nice feature of having a circular accelerators, and that is that we can run all these instruments in parallel. So we're running these instruments 24 hours a day, 6 days a week. We have 1 day for maintenance. And typically then a research group comes for a few days to run an experiment. And if I say 3 days, I mean 72 consecutive hours. So it's very hectic, but it's also a time when one can really focus on research. And then the following day, we have a new group of researchers, and this way we can serve sort of up to a couple of thousand individual researchers, external researchers per year. Now, 2 of our instruments are part of the remade project. We have Balder, which is an X-ray absorption spectroscopy instrument, where you can then look at chemistry, you can look at local coordination, local electronic structure, oxidation states, valencies, things like this. Very useful for studies on catalysts, catalytic processes, environmental science, and so on. The other instrument which I'm heading is called FORMAX, and this and that allows for multiscale structure characterization by combining X-ray imaging and X-ray scattering techniques. And I'll talk a bit more about that later. But a bit about the background of FORMAX, because it is a bit special. Those of you who have been to Sweden, and particularly those of you who have been to Sweden outside of the bigger cities, know that we have a fair amount of forest. Yeah, I think almost 3/4 of the area of Sweden is forest. So we have a lot of fantastic bio-based resources for new sustainable materials. There's plenty of academic research in this field. There's plenty of interest also from industry to develop new sustainable materials from the forest. And as a way of supporting this, private foundation, the Wallenberg Foundation, and paper and packaging industry have joined to finance an instrument at max 4 called 4max. And the agreement is then that with this funding both for construction and operation, then we can reserve 50% of our user time for research and development of new materials from the forest. And then the other 50% is available for general users. We've now been up in full user operations for a year, and yes, we are also part of the remade project. Now, if you're more interested in the background of 4max, then Physics World made a nice feature article a year ago or so. So you can find it by Googling. Okay, we're going to talk about multiscale structural characterization. And to set the scene, I thought I'd briefly introduce hierarchical materials, as we physicists like to call them. So many materials in nature and many not natural and synthetic materials are what they call hierarchical, meaning they have important structure, many different length scales that then govern some of the material's properties. Wood is a great example. Wood consists, among other biopolymers, of cellulose. And it's the ordering of cellulose at different length scales, sort of, that governs the the mechanical properties of wood. Now, if you look at those numbers there, we have sort of cellulose fibrils from nanometer length scale up to macroscopic structure of, let's say, 10 millimeters. That's 7 orders of magnitude in length scales. Now, if you want to understand the structure-function relationship in these kinds of materials, then we need some sort of multiscale structure characterization. And I'd say this is not limited to wood or other hierarchical materials. You could also have some processed materials that show heterogeneity, structural heterogeneity, and you need to understand this heterogeneity in order to understand the properties. A question then arises: how do we capture all these different length scales? And how do we maybe capture them also in situ experiments, perhaps, where one has an external stimuli? It's not so straightforward. Now, one way of studying sort of nanoscale structures or structure in materials that is fairly widely applicable to different kinds of materials is called small and wide angle X-ray scattering. So for those who are unfamiliar with this technique, basically we take an X-ray beam, we shine it on a sample, and then we measure a scattering pattern behind the sample. Typically, we then distinguish two different modes here. At very small angles, we talk about small angle scattering. And the figure there on the top right is small angle scattering from wood. Small angle scattering is, well, let's think about driving on a misty day. The light or the headlights from your car will scatter from water droplets in air, and you see this sort of halo around the headlight. That's small angle scattering. In this case, of optical light from fairly large water droplets. Now, if we go to X-rays instead, we see the same phenomenon. The difference is that now we're sensitive, or the scattering depends on the nanoscale density variations of the material that is scattering the X-rays. So that gives us a means for probing the nanoscale structure in these materials. At larger length scales, we typically talk about wide-angle X-ray scattering, and there we are in length scales, sort of where that we can probe also crystallinity of polymeric materials, for example. The image there, bottom right, is diffraction from cellulose diffraction from a piece of wood. Now, these techniques are a bit indirect in the sense that what we're measuring is essentially a square modulus of a Fourier transform of the density in the material. So typically one has to somehow model the structure and what kind of scattering pattern it would give rise to, and then compare to the experimental data to deduce the structure. But still, these techniques provide information on size, shape, packing, and orientation of nanoscale constituents in the sample. Typically sort of length scales from a nanometer to hundreds of nanometers. On some instruments, you can go up to a couple of micrometers even. And at modern synchrotron light sources, we go down towards milliseconds, fairly straightforwardly in the microsecond regime if one is doing some sort of pump probe experiment, for example. Okay, this technique has been around for a long time, has become more popular now with nanotechnology applications, and users have access to these instruments via the REMADE project. This is an example here of first REMADE experiment at MAX 4. We had the researchers visiting us who were interested in situ synthesis of carbon nanodots for water purification applications. Now, there is one caveat here, and that is that in an experiment like this, you're measuring some sort of statistical average over the whole scattering volume, the volume from where you're collecting scattering. And in traditional experiments, SWAXS or WAXS experiments, this is fairly large volume. So if you have a complex sample with many different components, or if you have, let's say, a processed material that shows structural heterogeneity, for example, then you average over all this variation, and the analysis can become quite cumbersome, if even possible. So a way to get around this is to carry out these kind of scattering experiments in a scanning imaging mode. So basically you focus down the beam to a small spot, and then you raster scan your sample through the beam, and then you different positions of the sample you carry out you do a sort of SWAXS or WAXS or SWAXS-WAXS measurement, and then you build up a map of the sample as shown sort of schematically here, where in each spot where you make an experiment, you're probing some characteristic size or for characteristic orientation of the sample in the nanoscopic length scale. And in this kind of experiment, then you're probing nanoscale features, but you then have a real space resolution of essentially of the size of your X-ray beam. We typically work with then a beam size of, let's say, a few microns to 50 microns, maybe 100 microns in some particular case, but it's in that ballpark. Micrometers typically, tens of micrometers. And then the field of view of your imaging experiment then is given by how much can you afford, how far can you afford to scan the sample. Now, you're still looking at a statistical average of the scattering volume, but in this case, it's much smaller. So you do sort of catch then heterogeneity or structural diversity on length scales larger than your real space resolution. And I'll show a few examples later of what you can do. So if we get back to this picture of wood at different length scales, with small and wide angle scattering, we then probe nanoscale features. In this case, sort of nanometer to a few hundred nanometers. But then because we do these experiments in a scanning imaging mode, we can extend this to mapping out larger samples up to millimeters, maybe even 10 millimeters of that order. So depending on your sample, but we can sort of then map out from, let's say, a nanometer up to 10 millimeters. That's these 7 orders of magnitude in length scales in a single experiment. Okay. What are then the limiting technical factors in an experiment like this? Now, for you who are not familiar with synchrotrons or accelerator-based light sources, there's a huge increase in the brilliance of the X-ray source when going from X-ray tubes that we can use in labs, home labs, for doing, for example, small angle scattering or diffraction. And then when you go towards the synchrotrons, the brilliance goes up by many orders of magnitude. Now, in the case of particularly small angle scattering, you need a fairly collimated, well-collimated beam. In the case of scanning SWAXS, you need a focused beam. And you need a fairly high X-ray flux because these are photon-hungry experiments. Basically, whereas you can do standard small and wide angle scattering experiments in a home lab, to the standard, then if you want to do scanning SWAXS, you do have to go to a synchrotron to have enough photons. So that's the first sort of technical limitation. Then another limitation is that these scans tend to become rather large. You have a small beam, you want to cover a large area. Beside the many photons that you need, you get at a synchrotron, you also need very fast detectors, much faster than sort of what Röntgen used 120 something years ago to image his wife's hand. Today, I mean, we typically run these with 100 Hertz or faster, these experiments. And then you also need fast stages for quickly scanning your sample to make use of the fast detectors and the high X-ray flux. So in principle, you can do these experiments at any SWAXS wax beamline, at any synchrotron, but in practice, some instruments focus more on more the traditional SWAXS experiments, and some focus more on scanning SWAXS wax experiments. A third technical limitation is if you want to do simultaneous scanning SWAXS and scanning WAXS experiments, then you need to have some custom detector, WAXS detector, so that you can pass through pass through the small angle scattering beam past the WAXS detector and onto a SWAXS detector. And those you don't have at every instrument. Now, if you want to do scanning SWAXS experiments within the remade project, then I would say, although the remade provides access to several SWAXS beamlines, practice there's only a few that are really suited for this. Formax at Formax, this is really one of the key techniques that we provide, so it's well suited for these experiments. A final comment is that that's all well and good, but in the end, it's radiation damage of the sample that often limits how good resolution you, for example, can have. So this is something that always needs to be discussed with instrument staff in advance, discussing means of mitigating radiation damage, for example. Okay, that was very briefly on the limiting technical factors. There's a nice preprint recent preprint by Christian Appel and his colleagues at the Paul Scherrer Institute that discuss several of these issues more in detail. If you're interested, I suggest you have a look at that preprint. Okay. Sorry. So if we then think about what kind of experiments we can do, we have a few different modes. We can scan the sample in 2 dimensions. Let's say transverse and vertical to the beam. So this is shown here as a schematic of sort of scanning SWAXS imaging on a piece of bone. And this is what I would call them SWAXS microscopy. You see that bone consists of mineralized collagen that exhibit anisotropic scattering in the SWAXS regime because the collagen have a preferred orientation. So by mapping the direction of the anisotropy, the degree of anisotropy of the scattering, one can then map out how the collagens are preferentially oriented within the sample, as shown then on the bottom left. And then one gets both sort of preferred orientation, degree of orientation in this case. One can extend that to SWAXS tomography. So one introduces also a tomographic rotation. And then one gets a 3D representation of some nanoscale structure within the sample. And again, this can be applied to both biological materials like in this image or material science applications. So in this image here, then in the middle group, research group from Denmark and Switzerland were looking at the concentration of neurons in a brain. In this case, the neurons are covered by myelin, which has a nice peak in the small angle regime, and then one can map out concentration of neurons in the brain. One can take that even one step further by introducing one more tilt angle, so a large pitch angle with respect to the beam, shown on the top right. And then one can do something called tensor tomography, where one is mapping out not concentration of fibers or fibrils or filaments in each in each voxel in the sample, but their preferred orientation, degree of orientation, and concentration. So this is some sort of, you could say, 6-dimensional imaging of the sample. You see on the bottom right, you see an example of the first papers where they were looking at local preferred orientation of collagen in a bone. But you can also apply it to other systems. On the top, you see maybe if you squeeze, you see that is actually a rat brain that is being imaged there, looking at neurons in a rat brain in 6 dimensions. Okay. Now an obvious drawback with doing sort of point-by-point scanning of to do imaging is that these experiments tend to be quite lengthy. So if you just do 2D imaging or microscopy, then it's still fine. We have fast stages. It's maybe a few seconds per line. If you take 100 lines in your image, then that's a couple of minutes. But if you want to do tomography, then you add 50 projections or so, then we're suddenly talking about a couple of hours, maybe. If you do tensor tomography, you do this at 5, 6, 7, 8 tilt angles, then we're talking about 10 or tens of hours. So as you can do, you can still manage to do SWAXS microscopy in 2Ds if you want to do something in situ. When you go to three dimensions or higher, then these are static experiments. At least today. Okay. I thought I'd give you some idea about the breadth of applications where these techniques are used. So this is now proposal statistics for the Formax beamline up to date. On the left, you see the distribution of techniques that our users are asking for. The gray area there is scanning SWAXS in some form. The yellow one is some combination of full field tomographic imaging and either SWAXS or scanning SWAXS, maybe with a side preference for scanning SWAXS. So a bit less than 50% of the proposal for the Formax beamline deal with scanning SWAXS imaging in some sense. To some extent, I think it just reflects that there is a big need for multiscale structural characterization. If you look to the right, then that's a distribution of the scientific fields or disciplines that our user base is working with. So given our sort of funding scheme, it's no surprise that roughly half of the proposals we receive are for some development of new wood-based materials. But then we have roughly 1/4 on life science, to a big extent the biomedical imaging, and that is to a big extent scanning SWAXS imaging. We have a bit of food science, interest from food scientists, and then we have a fairly large portion also of material scientists working on other materials than wood-based materials. And here the sort of scanning SWAXS applications range from in operando battery studies to aging of cast iron. So it's very broad. Sorry. Okay, a bit about the experimental challenges. And I usually like to present this as a map where on the horizontal axis, I try illustrate the effort and experiment needed for preparing an experiment. And then on the vertical axis, the effort and expertise needed to process the data. Now there are some techniques here that we don't discuss in this talk, but let's walk through the scanning SWAXS. So if you look in the top left, we have there sort of standard SWAXS experiments. And there, sort of, if you would start doing in situ experiments, then you need to spend more effort on preparing the experiment. You need more expertise to prepare that experiment. Now, if you start doing scanning SWAXS experiments as such, the effort needed for preparing the experiment doesn't change much. It's roughly the same. But the amount of data just becomes so huge that you need more and more effort and expertise to manage it. It really grows exponentially, the amount of data with increasing complexity of the scanning that one is doing. So whereas we can sort of still provide SWAXS microscopy as a sort of analytical tool that is fairly straightforward for users to carry out. Once you start doing SWAXS tomography or tensor tomography, these are really highly specialized techniques, and they're more of, I would say, expert tools. So essentially, if one wants to carry out these experiments, one needs some expertise in those techniques within the team of researchers carrying out the experiment. Now, for many of our users, that means that they collaborate. That is the key to accessing these kind of techniques. Now, luckily, Remade can help with this, since one can get support from our junior scientists via the Smart Science cluster, and they can serve as sort of an expert that helps users with planning and analyzing, planning the experiment, carrying out the experiment, and sort of analyzing the data. Now here I just want to mention Santiago, who is then the junior Remade scientist working at MAX4 and at the Formax beamline. So if you're interested, please contact Remade, and maybe Santiago is the one who will support you. Okay, I thought I'd finish up by a few examples just to give a bit of flavor of what kind of experiments one can carry out when doing some sort of SWAXS imaging. So this first example here is in situ assembly of bio-based nanoparticles, in this case cellulose nanoparticles. Now this is an older experiment, so it was not carried out at MAX4 or the Formax beamline, but this research group from Stockholm and by Daniel Söderberg is carrying out experiments with us nowadays. So their interest was, among other things, to sort of assemble nanocellulose suspensions into gel fibers with very good mechanical properties. So they have these sort of microfluidic chips where they flow the suspension, they can focus the particles there, and then they can follow a function, but by scanning SWAXS, they can follow as a function of position in this chip how the orientation of the particles change. That's shown here in, let's say, in the C panel, how one order parameter then is sort of changing, and they can then estimate where they have a maximum orientation and where it would be. And they and then with this kind of data, they can tune the flow and addition of ions and so on to cause assembly of the particles into fibers. Now these are still sort of fairly sparse measurements, but today with the intensity of the X-ray beams and fast stages, one can really map out into the ordering of nanoparticles in such microfluidic channels. Sorry, and then they can they form these fibers then that have rather impressive mechanical properties, especially considering that it's I don't know 95% water maybe. Another example is looking at processing of materials, and this then is a very applied example. This is done in industry all the time, but the processing does induce heterogeneity of the samples. So this is now example of injection molding of polyethylene films in this case. So not exactly directly relevant for the remade project, but I think it's a good example of what kind of scanning SWAXS experiments one can do. So in this case, the group then would make samples with different injection molding process settings. They can then characterize the structure or variation of the nanoscale structure along these films, different positions of the film. This could be, for example, how the degree of orientation varies across the film in different positions along the flow direction. Concentration of polymers along the and so on. One can take these kind of data and compare them to virtual modeling of the manufacturing process, sort of to validate the numerical or virtual modeling. And once one has validated the modeling, then one can use that as a tool for improving the process. Now, this particular group also carried out some experiments at Formax. So this was a packaging company, Tetra Pak, together with Marianne Liebe's group from the Paul Scherrer Institute. And they also worked at Formax on sustainable paper straws. So there's a need to use more bio-based raw materials in food packaging, for example, paper straws being one example. And then one needs to improve and optimize the properties. So it's this example here on the bottom, and this is still unpublished data, but in this example here, the group then studied the corrugated parts of a paper straw, where the paper straw was manufactured in two different processes, an older one on the top and a new one on the bottom. And in this particular case, they were looking at some nanoscale defects in the paper straw due to the corrugation process. This group can then take this kind of data again. They can compare it to their virtual modeling of the manufacturing process, and perhaps then find means of improving the process and then improving the product. Now, as I said, this has not been published yet, but we are very proud that Tetra Pak decided to highlight this collaboration in their sustainability report from a year ago, showing how important it is also for companies to use these kinds of techniques for their R&D efforts. Okay, I'm coming towards the end of my talk. Masood already mentioned about deadlines for getting access to do experiments, and the next proposal deadlines are during this month and October, depending on the mode of access. Finally, I just want to highlight a workshop that is coming up in the end of September in Lund, zooming into bio-based materials, which is then organized by Max4, Remade, and Research, which is a platform for Swedish research platform for research on bio-based forest-based materials. And this workshop will focus on sort of the journey from a first-time user at synchrotrons to an experienced user. And hopefully it is very useful for those who are interested in looking into SWAXS and imaging experiments at synchrotrons. So the deadline is on Monday. You still have time to register if you are interested. You can find links on the homepage of Max4. Okay, with that I come to the end of my talk, and I'm happy to take questions.
Thanks, Kim, for this nice and didactic presentation for our general audience. So, yeah, meanwhile, we are waiting for questions. I have one. My question is about the sensitivity of the technique when it comes to diluted systems. So let's talk about, let's say, inorganic system, let's say semiconductor or semiconductor oxide, doped semiconductor. So in this case, what would be the let's say the sensitivity for probing clusters, for instance, doped clusters in the semiconductors?
Yeah, so sensitivity, it's two things. So first of all, in general, if you think about small angle scattering, I didn't mention here, but the scattering power then, if you have a suspension of nanoparticles, scales like the radius cubed. So the larger the scattering constituents, the stronger the signal. And that's a pretty strong dependence on the particle size. Now you can still study small particles, but then you need a good contrast. The contrast mechanism then, contrast is essentially then given by differences in electron density. So let's say metals obviously have a stronger or a higher electron density than organic matter. They tend to scatter more. To give you some, now the SWAXS instruments, at synchrotrons, they do optimize the instruments sort of to be able to measure also samples with low contrast. So just to give a sort of an example, at Formax we have we can measure, let's say, cellulose nanoparticles in water at concentrations of well below half of 8%. And if you look at the contrast between cellulose, that is carbon, oxygen, and hydrogen, compared to water, then it is rather big. So you have good sensitivity when you go to the synchrotron instruments. At lab sources, yes, perhaps not quite as good sensitivity.
So if there is no other question, then seems not. I want to thank you again for your nice presentation.